Although exact numbers are not known, it is thought that millions of animals are killed during chemical safety testing every year. This presentation will explore how biological substantiation using tools such as high-content omics technologies can strengthen chemical grouping and read-across approaches, a non-experimental predictive method used in chemical safety testing, with the potential to reduce animal testing and animal death. Chemical safety testing is vital for the preservation of human and environmental health. An acute example of the importance of chemical testing is the elixir sulfanilamide tragedy of the 1930s, which accelerated and formalized regulatory toxicology testing, and more currently this is reflected in the growing general awareness and concern regarding so-called forever chemicals. The human population is exposed to a high number of chemicals. In Europe and America alone, hundreds of thousands of chemicals are registered for use with chemical regulatory bodies. However, only for a percentage of these chemicals has sufficient information enabling proper chemical safety assessment been provided. Regulators' reliance upon animal-derived toxicological data can partially explain the significant data gap. Furthermore, it has been suggested that it is not feasible to fill this data gap under the traditional animal testing-based paradigm, not only because of the tremendous number of suffering animal subjects that would be required, but also because of the associated resource and time cost. In Europe, the REACH chemical legislation requires manufacturers and importers of chemicals to provide physical, chemical, toxicological, and ecotoxicological information upon chemical registration. This data must be gathered through appropriate specified tests or acceptable alternatives and must be used to assess the safety of the chemical. For example, toxicological information requirements can include information on lower-tier endpoints such as skin irritation and higher-tier endpoints such as repeated dose toxicity. Importantly, however, REACH legislation does provide the opportunity for these standard information requirements to be provided through non-animal testing alternative methods. These options, which allow the waiving of traditional tests and the conditions predicating their use, are listed in Article 13 and Annex 11 of the REACH legislation. One of the most developed of these alternative methods is the chemical grouping and read-across approach. ECHA have developed specific guidance for this approach, the read-across assessment framework, and it is the most commonly used adaptation and therefore reduces the need for further animal testing. Chemical grouping and read-across is a data gap filling method which relies on the assumption that similarities between chemicals in structure reflect similarities in chemical toxicological effects, and therefore the toxicological effect associated data which is available for existing chemicals may be read across to a structurally similar investigated chemical. Satisfying the standard information requirements for specific toxicological endpoints and eliminating the need for further animal testing. Structural similarity is a prerequisite for acceptance of chemical grouping and read-across approaches. Registrants must provide a clear read-across hypothesis stating the structural similarities on the basis of which a chemical group is proposed. Registrants must justify their hypothesis and explain why the toxicological endpoint properties for the investigated target chemical can be predicted from the structurally similar source chemicals in the proposed chemical group. Tools such as the OECD QSAR toolbox are used by registrants to search databases containing existing information on the toxicological effects of particular chemicals for structurally similar source chemicals based on chemical descriptors of the target chemical. Toxicological endpoint data can then be read across from source chemicals deemed sufficiently similar to the target chemical and can be used to fill in missing endpoint-specific standard information requirement data for the candidate chemical prior to registration. Despite the promise and frequent use of this approach, in reality, read-across proposals are often rejected by ECHA due to shortcomings in the hypothesis and justification. Furthermore, it is known that structural similarity does not necessarily lead to similar toxicological effects. Among known examples of this are the highly structurally similar liver enzyme inducers, acetylaminofluorine and 4-acetylaminofluorine, the former inducing tumor formation and the latter in fact being non-carcinogenic. As chemical toxicity is the product of complex biological mechanisms, mechanistic similarity is among the non-structural properties which can strengthen a read-across proposal if demonstrated as similar between source and target chemicals. In fact, an importance of mechanistic understandings of chemical toxicities is reflected throughout chemical safety testing and in the collaborative development of adverse outcome pathways. Adverse outcome pathways can be used as pragmatic, simplified characterizations of the biological mechanisms involved in chemical exposure-induced development of toxicological adverse outcomes. They characterize and consist of the chemically induced molecular initiating event, the subsequent chain of key events occurring at different levels of biological organization, and the resulting toxicological adverse outcome. It has been suggested that chemicals could be grouped by their ability to trigger the same molecular initiating event or downstream key events. Mechanistic similarity could be assessed for and demonstrated by a similarity in generated in chemical or in vitro data characterizing potentially shared molecular initiating events or key events. However, the very small number of regulator-approved adverse outcome pathways, and therefore the lack of chemical space that they can currently cover, means that a mechanism-based substantiation of chemical grouping and read-across using adverse outcome pathways is a distant prospect. If a mechanism-based substantiation of chemical grouping and read-across is a distant prospect, perhaps a more reachable intermediate approach can be used. Patlowitz et al. In their 2015 paper ask whether it is always necessary to have data that provides a mechanistic insight into toxicity to support read-across, or whether a more general biological activity approach could be utilized. Could determining similarity in general biological activity provide value in strengthening chemical grouping and read-across proposals based primarily on structural similarity? If so, to this end, Patlowitz et al. Further suggests the use of biological profiling tools such as transcriptomics and metabolomics. Omics technologies are high-content tools which can provide a comprehensive snapshot of the global molecular activity occurring across multiple biological pathways within a tested system. Transcriptomics technologies use techniques such as microarrays and RNA sequencing to characterize and quantify RNA transcript levels in the test system and are a more mature and developed technology. Metabolomics technologies use techniques such as NMR spectroscopy and mass spectrometry to characterize and quantify metabolite levels in the test system, and thus provide a closer reflection of phenotype, but this field is less well developed. In the context of chemical safety, these relatively recently developed omics technologies allow investigators to conduct thorough molecular level interrogations at a previously impossible level, shedding light on the black box of biological activity which underlies and precedes the manifestation of toxicological endpoints. In recent years, transcriptomics and metabolomics technologies have been investigated in a variety of contexts and systems to determine if they can provide a reliable biological activity-based characterization of chemically induced toxicological outcomes with a predictive potential, which could be used to biologically substantiate and enhance traditional structure-based chemical grouping and read-across. The general process for this omics-based biological substantiation of chemical grouping and subsequent read-across involves firstly The measurement of differential gene expression or metabolite levels on target and source chemicals exposure to generate so-called gene expression and metabolite fingerprints for each chemical. Secondly, the calculation of the similarity between these chemical exposure fingerprints. And finally, the use of the tool to visualize the omics-derived biological activity-based chemical grouping. This biological-based chemical grouping is then compared with the traditional structure-based chemical grouping to either substantiate or refute the grouping and therefore the subsequent read across. A key example of this type of study was conducted by Ravensway et al. In 2016. In this proof-of-concept study, using 28-day rat in vivo metabolomics data, the group attempted to biologically substantiate a structure-based chemical grouping of phenoxy herbicides in order to read across 90-day repeated dose toxicity data. The omics data showed very similar metabolic changes between source and target chemical exposure and biologically substantiated the structure-based grouping. In fact, the results of the actual 90-day studies agreed with the read-across data substantiated with metabolomics data, demonstrating that using this approach, the 90-day data could indeed have been waived, and also demonstrating the utility of omics-based biological substantiation of chemical grouping and read-across. Furthermore, a great leap forward took place in 2019 when a follow-up study by researchers in the same group used the same method with different chemicals to submit a real read-across proposal to REACH. The results of this proposal will be a key milestone in the field and provide valuable feedback for future registrants. The revolutionary effect that omics technologies have had in many scientific fields has not been reflected in regulatory toxicology, not only in its potential to enhance chemical grouping and read-across, but also in the many other roles it could potentially play to improve regulatory toxicology and, in so doing, help to reduce further animal testing. There are a number of reasons for this, and some of these stem from the inherent complexities and perhaps suspicions that accompany any non-standardized, continuously evolving technology and methodology which relies heavily upon expert judgment and which produces data which can be interpreted to produce greatly varying conclusions. Varying solutions have been proposed to overcome these barriers, and because of the sizes of the workflows associated with omics methodologies, there are a number of different steps which could be worked on. However, perhaps it makes sense to gain momentum by focusing on the lowest hanging fruit. This has been suggested to be the adoption of a standardized reporting framework for any transcriptomics and metabolomics data submitted to regulatory bodies. In fact, this very approach is being tried currently by the OECD in the development of a hybrid transcriptomics and metabolomics reporting framework. And it will be interesting to see the effect it has on the value regulators place on omics data provided by registrants. Conclude, any paradigm shift surely requires a significant amount of time and concerted efforts to develop a better, viable, and equally effective alternative to the status quo. The move to true animal-free chemical safety testing is no different. However, progress is being made and is being facilitated by ideas and technologies such as featured in this presentation. Thank you very much for listening.